It's a whole different world out there. Like the olfactory subgenome is just so different from person to person. It's like a little brain sitting right here that's constantly deciding what are the inputs that go into your brain. Our noses, the human noses, they're packed with all of these sensory neurons. We call them olfactory sensory neurons that express a family of 400 different olfactory receptors. Each neuron will express only one receptor. And the way they function is anytime that you breathe in, all of the smells from the environment are going into your nose and they hit the receptors. So if a receptor is recognizing the odor molecules, it's going to bind it and then send a signal to the brain that tells the brain that the receptor is seeing a molecule. And it's complex. It's all pretty complex because, like it's a limited set of 400 receptors, and that is being used to detect and discriminate millions of different odors from the environment. So for example, when you're smelling coffee, a particular subset of olfactory receptors are going to get fired. And when you're detecting smelling a perfume, then a different a subset of olfactory receptors are going to get fired. Now there may or may not be some overlap depending on how similar or different the odors are. But you can imagine that using this sort of a combinatorial mechanisms of receptor identity and intensities, relative intensities, even though the number of receptors is limited to 400, the possible combinations are practically infinite, right? And using those infinite combinations, like the human nose is empowered to detect and discriminate so much that is going on in our chemical environment.